Since the 1800s, society has looked for ways to lose weight, like drinking water mixed with vinegar, adding a grapefruit to every meal, or eating soup made from cabbages twice a day for 2 weeks, which really just seems like a great way to create a lot of gas. We've also developed and followed all sorts of diet plans: Atkins, South Beach, Mediterranean, Paleo, Keto. At the same time, we've developed and followed a whole host of exercise routines, each designed to target specific muscle groups. Things like endurance, circuit training, powerlifting, yoga, Pilates, hula hooping, even walking and running backwards. At times it feels like we've fallen into some sort of weird blend of science fiction and urban myth. What? Wait, what? Whoa! Anywho, we used to think that losing weight was a simple matter of eat less and exercise more. Now we realize the equation is much more complex and is impacted by the amount and type of exercise we get, the food we eat, even the type of bacteria that's present in our gut that extracts more or less nutrients from what's in our diet. Data now also suggests that our genes may impact the success of our diet and fitness plans. A whole host of companies have developed a new set of genetic tests that they claim will allow you to identify the fitness and food plan that will optimize your health. Hey Elvis.
How you doing, baby?
Peanut butter, banana, and bacon sandwich?
Oh, thank you. Thank you very much.
So can genetic information transform our quest to stay in shape and lose weight? That's a topic we'll explore in this session. How's it taste?
Just like Mama used to make.
Excellent. Good evening. Welcome to our third week of the 2018 Biotech 201, How Can Biotech? We have quite an interesting evening ahead of us tonight, as you can see by the props on either side of the stage. We'll be focusing tonight on how can DNA help me lose weight and predict my death. And there's a cookie break in the middle. So yes. This is going to be, there are so many things that we could talk about tonight, and I am going to sadly just touch on a number of things. Like skipping a rock across a pond, we're not going to be able to dig deep. We're just going to dip into lots of pieces. I apologize in advance that we're going to move through some of this pretty quickly, but I want to give you a sense of the landscape of where this stands. So there are a whole host of companies out there that will offer, this is 20% off your genetic workout system. Or this one, discover the diet plan built specifically for your DNA. Can DNA testing help you find a better workout? And the truth about DNA-based diets. So before we dig too deeply into any of the topics tonight, let me tell you a little bit about how science is often done, especially in the field of genetics and genomics. We can start when we're trying to solve something. We can start by saying, all right, I think this gene, based on what I know about this gene, should be involved in this set of symptoms, should be involved in weight, because this is a gene that's involved in fat metabolism. So I'm going to analyze, I'm going to test whether changes in this gene are found in a population that has a set of overweight or underweight individuals. That's hypothesis-driven science. I have an idea that I'm specifically testing. We also have hypothesis-independent science, where I don't have any prior knowledge or any expectation of why I should be looking at something specific. As the field of genetics has reached a point where I can now reasonably sequence hundreds of people, and I can look at millions of places, thousands of places in their DNA, I can now ask at any one place, do I seem to see an association between a change at this site and this set of symptoms. A change in this site in DNA and issues with obesity, for example, or with type 2 diabetes, or with Alzheimer's. That's independent, hypothesis independent. I'm just essentially doing the equivalent of an enormous fishing expedition because the science now lets me look at all that information. Genomics is empowered. We can now do hypothesis independent work because of the technology. But what that means, and you will hear this tonight, is that we often have associations where we don't understand the biology. So, yep, there's a link here in some way, shape, fashion, or form, but I can't tell you why. And so I don't know what that means going forward. The science has to catch up with that. The benefit of that is that means if I truly find a real set of genetic signatures, for example, a set of genetic signatures that helps me early diagnose cancer, if I don't understand the biology, but I know it's a firm connection, that's an incredible early diagnostic tool. On the flip side, I may find an association and suddenly everybody starts treating it as if it is gospel, and then we discover later on that that was just a one-off that I couldn't replicate in any other studies. So those are two sides of the kind of data that we get from hypothesis-independent research. And I just want to give you that background because I don't want you to walk away from this thinking that scientists just willy-nilly pull data out of the air and say, here, look, we've got this finding. I just want you to kind of understand that background. Everybody with me on that? Makes sense? Okay. All right, so I'm going to tell you about the kind of genesis of genetic variants that could be associated with weight loss and the story that brings us why we're having this conversation. Now, I'm going to tell you just one of lots of stories, so I could focus on lots of other genes. I'm just going to follow one particular storyline all the way through. So this actually starts in 2007 with a study that was done with about 300 overweight postmenopausal women. And these women were randomly assigned to one of 4 different diets, and they followed this diet for a year. These are the diets: Atkins, Zone, Learn, and Ornish, and you can see out to the left of them a little bit about them. So Atkins is a low carb, Ornish is a very low fat, those sorts of pieces. So these 311 women were assigned to one of these 4, randomly assigned. They were given coaching and support throughout the entire year, and there were all sorts of metrics taken throughout the year. And at the end of the year, the amount of weight they lost or gained was assessed. And this was the average of those groups of women, the amount of weight that was lost. Now, again, this study was published in 2007 in the Journal of the American Medical Association. And this study, which was a well-done study, suggests that, number one, that Atkins, very low carbs, seemed to do better when you were ranking these. But the really important piece that didn't make the headlines was this. Within each diet group, there was a 40 to 50 pound range. So you had some of these women that lost 30 or 40 pounds, and you had some of these women that gained 10 or 15 pounds within every single category. Some of you are going, yeah, that would be, yeah. No judgment in this room. So some of these women, regardless of what diet they were on, lost huge amounts of weight, and some of these women gained weight regardless of what diet they were on. So, this is really interesting. Okay, yes, Atkins looks like you lose more weight, but there's huge variability. Which led to the question, why do some individuals seem to do really well on certain diets, and some individuals not do well on the exact same diet? So the authors of this study then did a follow-on study. As they had recruited the women into the study, they had samples of their DNA, so they could go back and test the DNA. And so in 2010, they published a follow-on study where they took a subgroup of these women, 133 out of that original study, and these women had already gone through that diet year. They didn't bring them back in to do more work. They just analyzed them because they also had some genetic information. And there were 3 genes that they looked at, and you can see them on the screen behind me, and we'll dig into them in just a minute. But they looked at changes in these 3 genes that other scientists had suggested changes in these genes might be associated with how well people would do on diets. So they analyzed these 3 genes, one change in each gene. So they looked at 3 different genetic markers, and they said, do you have this or do you have this at each of those 3 markers? And as they did this analysis, they came up with what looked like specific genetic combinations that might suggest these women would do really well on a low-carb diet. Their metabolism would be most sensitive to cutting out carbs, or a low-fat diet. Their metabolism would be most sensitive to cutting out fat, and a small number of women that kind of had an indeterminate result that suggested maybe a more balanced diet. So they then looked at at these women and said, okay, if we break the women into groups based on what diet were they on, and was that diet matched to what their genetics actually suggests would have been an ideal diet, do we see anything different? So I'm going to make sure I make this really clear. These women had already gone on this diet for a year, and now they went back and they looked at their genetic information and said, oh, look, this woman looks like what would be an ideal diet for her is low fat. Was she actually assigned to the low-fat diet, or was she assigned to the low-carb diet? And what were her results? And so this is the information. This is what they found. So let's just focus on this section on the left. The women that were on what's called the genotype-appropriate diet, so what their genetics suggested would be the right diet, was actually the diet they were assigned to in the study. They lost an average of 13.2 pounds. The women who were not matched, so their DNA suggested they would do well in this kind of diet, and that was not the diet they were put on, they lost an average of 4.6 pounds. So 2.5 times less weight loss when they were not matched to their DNA diet plan. And then the same sort of thing when they looked at how many inches were lost. So this, you can go back and look like 2010 when this study was published, this gave rise to your DNA will tell you what diet you should be on. There is an ideal diet plan for you based on what your DNA is telling you. And if we can look at your DNA, specifically these 3 markers, we can tell you if you should be on a low-fat or a low-carb or some other type of diet. Gary?
Were there any twins in this study?
Ooh, were there any twins in this study? I don't know. I have no clue. Where you're going, they would have identical DNA, and you would think that they would then both be responsive to the same kind of diet. You would think.
But sometimes they're not.
But sometimes they're not. That's right. That's exactly right. So this was huge news. This confirmed what intuitively people felt, that my DNA, my body, is designed for a certain kind of metabolism, which may not be the same from person to person. And if I could just align my diet with what my DNA says, then weight loss is going to be so much easier and so much more targeted. And there are now a whole host of companies that will look at these 3 markers, 50 markers, 100 markers, and say, you should be on this kind of diet, you should be on this kind of diet, you should have this kind of exercise plan, your genetic information suggests you should really do interval training, your genetic information really suggests that you should be doing high endurance training. You should be looking at this kind of a sport, you should be a sprinter, you should be a marathon runner, you should be a weightlifter. You'll do best if you have this kind of diet followed by this type of exercise plan, followed by this type of vitamin nutraceuticals. Did they control for activity level? In this study, my understanding is they actually were able to control. They were able to track the women's activity level. They were able to actually track what they said they ate. So they tried, and I'm going to come back to that point because that's a really important point in just a second. So what are these 3 genes? I'm not going to read this to you, but I just wanted you to have it. So here are the 3 different genes in black. We use lots of big words for these genes. And then in red are some of the key features about what we think these genes do and what we think these changes might do, how they might impact diet and weight loss. And you can see lipid storage, fatty acid uptake, the release and use of fatty acids from fat cells, how we break down fat. This biologically, it makes sense. It's plausible that changes in these genes might influence how your body would deal with fat storage and fat breakdown. And if we had held this class last Tuesday, I would have wrapped this question up by saying, and so this is now what a lot of groups are looking at. And there are people that question how well that works, and clearly not everybody is going to fall into, I'm going to look at 3 places in my DNA and determine what my ideal diet is, and then I would have moved on to the next set of slides. But last Wednesday, I tell you, this field moves. This field moves fast. This is why every year we have a different 201. Last Wednesday, a follow-on to the follow-on was published by the same group out of Stanford. And in this instance, they looked at 609 overweight adults. And their goal was to say, can we replicate our prior findings? So critical. You have heard me say over and over and over, one finding is really intriguing. We have to have it replicated, and we need to have it replicated more than once. So 609 women. They were randomized to a year of low fat or low carb. These women had weekly initially, and then every other week, and then every month meetings with counselors talking about how you plan your meal, the selections that you make when you go out to eat, how you avoid emotional eating. So these women got a great deal of support throughout the year. These women tracked at random times. They tracked what they ate in a 24-hour period. They tracked the amount of exercise and activity they got, all sorts of information. 79% of these women completed this year-long trial. This was an intensive trial. And they also looked at their genetic information, and they looked at some other pieces as well. And at the end of the study, this was the average weight loss for low fat and for low carb. So not really that much difference, not statistically significant in difference between were you on low-fat diet or were you on low-carb diet. So then the question was, okay, were the women whose diet was matched to their DNA, did they do better? Because the prior study would have suggested, if that held up, the answer was yes. No. No difference if you were matched, genetically matched to your diet type, or not matched to your diet type. This got a whole lot of press in the last week, including this article with the headline, "We didn't even come close," the researcher says. This is a significant finding. This is a well-developed study that asked all the right questions, that set everything up the right way, that really thought they were going to get an answer that confirmed what their previous study had found. Not at all. So they went back and said, why did we get the finding, our earlier finding? Okay, none of us in this room are robots. None of us can easily be controlled. And life happens. In the middle of everything else that we plan for, life happens. So it turns out in the 2010, the women that were analyzed in 2010 but actually were part of the diet in 2006 and 2007, a number of women that were in the group where their genotype, their DNA, matched their diet, a number of those women were incredibly motivated and meticulously counted their calories and did so well and lost so much weight that it made the DNA matched my diet group look really good. And a number of women in the non-matched groups, just by chance, because remember these were randomly organized, randomly put into these groups, a number of non-matched women had major life events like the death of a spouse or the divorce of a divorce from a spouse or some other thing that totally wreaked havoc on their diet. And so that as a group made the non-matched look really bad. So it wasn't the DNA, it was by chance you had a whole group of super achievers here and a whole group of life happened and threw me off here. That sounds familiar. Every single one of us have been in those places. But I think it talks about the challenge of any kind of large-scale study, whether we're talking about genetics or anything else. How do you control for all of these variables in a world where life seems to happen in very random ways? So, does this mean that there is no impact of our DNA on our diet? I think the answer to that question is no. I think, and many scientists think, that there are clearly genetic influences on how we metabolize energy, store fat, and break down fat. We know that there are these genetic differences because we see very rare genetic mutations where changes in a specific gene impair somebody's ability to store fat, or to break down fat, or to metabolize energy in a certain way. So we know there are genetic factors. But to think that a 3-gene test is going to say, here's your match, I think is, in hindsight, probably incredibly simplistic. So it's likely that there are multiple factors. Those of you that have been through either the microbiome study we did for 201 or any of our more recent Biotech 101s when we've talked about the microbiome may recognize this image. This talks about all the different ways that potentially we end up increasing adipose tissue, increasing fat storage. We can do that through a whole host of different ways. Pregnancy, normal physiological changes, changes that are because of my diet, because I have more caloric intake and less caloric burning. Changes because of my bacteria. There are certain types of bacteria that do a better job of extracting nutrients from the food that we eat than other types. So it's this combination, and there are genetic factors as well. So scientists are now beginning to think, okay, maybe what I really need to be doing is I need to be looking at hundreds or even thousands of genetic pieces and trying to create a pretty sophisticated algorithm, because each one of these genetic factors probably contributes a relatively modest event. So, take-home point from this is not that your DNA doesn't help you lose weight or harm you in an attempt to lose weight, but we aren't yet at the place where we can say, this one, this one, this one, and this one point you towards this kind of diet. Now, that doesn't mean that there aren't groups out there that will tell you, I'm going to use your genetic information, and at some point, I think we're going to get there. I truly do. And literally, if this session had been last week, you'd be hearing a somewhat more nuanced story out of my mouth. I mean, you know, I opened up my computer and started doing literature searches on Wednesday morning, and I said to my wife, wow, I have completely just changed the whole focus of what I'm going to talk about for one of the stories next week based on this paper that has just come out. Which also tells you science is continually adding to our body of knowledge. All right, along those same lines, that was diet. And it would be great. You see, I've got this great mannequin over here. I know that all of us would like to be able to figure out, or many of us would like to figure out, how do I just dial it in? Can we gene edit me and make some changes? You know, how do we do that? We are not at that point. So do not take last week's CRISPR conversation and try to tie that into this week's and think, well, I'm just going to gene edit my way to a size whatever. In the same way, we can ask questions about, are there specific exercise plans that should be optimized based on my DNA? And you can find companies that will offer you that. So in 2015, the British Medical Journal actually published a consensus paper where they addressed this, because there were groups that were actually offering tests to parents of young children to target the most likely sport that your son or daughter will excel in based on their DNA. So this is what you should put them in. So here's the key piece. The consensus among sports and exercise genetic researchers is that genetic tests have no role to play in talent identification or the individualized prescription of training to maximize performance. Consequently, no child or young athlete should be exposed to direct-to-consumer genetic testing to define or alter training, or for talent identification aimed at selecting gifted young individuals. Same kind with a slightly different twist on, are there specific nutritional foods or vitamin plans that I should take? Because there are a lot of companies that would love to look at your DNA and tell you, here's a set of customized nutritional supplements that you need to buy from us, designed just for your specific genetics that are going to maximize your performance. So this one looked at multiple different studies examining, is there any evidence right now that your DNA, that we know what DNA variants should say you should take this nutraceutical or that nutraceutical. Solid scientific evidence is currently lacking and we cannot recommend it. But here's an important caveat. Notwithstanding, in spite of that statement, the need for a thorough and continuous nutrigenomics research is evident, as it is a highly promising tool towards precision medicine. In other words, there's enough evidence, early evidence here, that this is likely going to be important. We each metabolize food in different ways, and we each have different things that we respond to. So at some point, as the scientific evidence builds, this may become really quite valuable. Again, sometimes I feel like a broken record with y'all, saying over and over, the science is not there yet. We get closer every single year, but the science in these instances is not there yet. All right, let me stop, because we've got a chunk of time that we can take questions. And those questions can be from what I've just talked about, or given how much information I ran through last week about gene editing that we didn't get to ask questions on, they can be on that as well. So questions from the crowd or questions from our online audience as well. Mark.
Since you opened up the topic of gene editing, can you talk a little bit about where the state of the art is in terms of using gene editing on cancer cells and targeting the areas of cancer cells that cause them to be cancer cells?
Yeah, so Mark's question is, can I talk a little bit about what is happening in gene editing as it relates to cancer cells? So we talked a lot about gene editing as it relates to trying to treat disease, and I only just glancingly touched on cancer work. A lot of the clinical trials, in fact, in the United States, the first gene editing clinical trial is going to be a cancer trial. And it is aimed at inactivating a specific gene or two in the immune cells of a patient with cancer, where this gene is known to make the immune cell essentially unresponsive. It makes it go, it's the equivalent of going nose blind to an aroma. The immune cell no longer recognizes the cancer, and so this gene editing actually silences that gene, so the immune cell hopefully now recognizes and revs up its activity. Many of the immunotherapy pieces that we seem to be hearing more and more about, especially the really expensive immunotherapy, are built around the idea of doing gene editing. So you could imagine that I have a massive cancer cells, and one thing we've talked about before is that in a tumor, not every cell is genetically exactly the same. It's what's called heterogeneous. It's got multiple mixtures of mutations. You can imagine a gene therapy, a gene editing tool that goes into cells and actually damages, cuts the genes that have a specific cancer-causing mutation. You could also imagine that could be used in precancerous cells to take precancerous cells back to a normal rate. Lots of work in cancer right now in the lab, in cell lines, and in model systems, just starting to see it make its way to patient therapy. So Lori asked if I can talk a little bit about metabolism. All right, let's see if I can find it. Yeah, that's it. This is actually the name of the journal that the article was in, because it was a microbiome article. And so you're now going to find that I can only talk barely about metabolism without diving into a place where I'm way in over my head, especially as it relates to the genetics of our metabolism. So rather than have me tap dance around that, I'm just going to say, no, I can't. Stump the teacher.
Relative to the first question, last week in NPR they were talking about some of them have experienced, like you said, the stopping of the progress of the, yeah, okay. My question is, Hudson Alpha, do you guys have any international exchange relative to this kind of research, or we are solo?
Oh, does Hudson Alpha do international exchange around this type of research, or do we go it alone? So broadly, there's almost nothing at Hudson Alpha that we go alone on. The field is driven by collaboration, at least from an academic perspective. You may get a different answer from a company, an intellectual property perspective, but the fields are driven by groups working together and bringing in expertise. So much of our work actually collaborates with scientists in other places around the country and actually with other places around the world. Our gene editing work is still relatively young and relatively new and is really aimed more at proof of concept kind of concepts. We do some collaboration. Dr. Eric Mendenhall, who's an adjunct faculty member at Hudson Alpha, but also at UAH, does a lot of CRISPR gene editing kind of work. We're doing some gene editing work in Dr. Meyer's lab, exploring ways that we may inactivate Huntington disease causing mutations. And that's in collaboration with a couple of other Huntington's groups that we're working on. So there are collaborations here around CRISPR. Now, your point about China, the NPR story, that there are lots of clinical trials going on in China, almost 100 clinical trials in China. And there are clinics set up specifically to run these gene editing clinical trials. The rules and regulations are slightly different in China. I should probably just stop that right there. So we'll just end that sentence. We'll just right there. Gary, you had a question?
Well, I was just wondering about these, the sports, direct-to-consumer genetic testing for predicting sport performance and talent identification. That was only the genetic testing. There was no environmental piece to that, meaning that the children or whatever, they might, yeah, sure, this particular test might tell them that it might go in this direction, but from their friends and their environment and everything else that they're doing, they might go in a different direction. That's why that might.
Yep. This comes back to the whole concept of athleticism is a nature-nurture. Sports talent is a nature and a nurture. Surely there are some genetic factors that may predispose you to playing well, to biologically doing well, physiologically doing well in certain sports. But your love of a sport, the reason why you play a sport, the amount of practice you put into a sport, plays, I would imagine, an even larger role. You may have to overcome more because you are genetically hindered or you are genetically advanced. But it has a much broader role than that. Other questions? Yes, ma'am.
Go back to Mark's question from last week. I believe the chart that you had that talked about the gene editing on the cancer had 3 types of cancer: myeloma, sarcoma, and melanoma, which are all very bad prognosis kinds of cancer. Is that why they were picked, or was there something specific about those 3 types?
So the question was the slide that I showed last week about the cancer and the gene editing of cancer talked about myeloma, sarcoma, and melanoma. And why were those chosen? So I'm going to take a slight at the edge of my knowledge, but it is likely that the subsets of those cancers were chosen because they had specific known mutations that could be good candidates for gene editing. So at this stage, it probably was not that they were chosen because they had such poor prognosis. It probably was that they were targetable. They were the most easily targetable, and they may have the fewest other therapies around. So it's probably a combination of the two for why those types are some of the initial ones chosen. All right, 7:07. We're going to go to break a little bit early. We're going to take about a 10-12 minute break. I'll be up here answering questions. As in our previous weeks, we've also got a genetic counselor who's on hand. If you've got questions of a family history nature, she'll be up here as well. Enjoy break, and I'll see you back in about 12 minutes.